I'm Hayley. Tonight we have a special discussion exclusive to UWA News. First off, the C value. What is it? It's a measure of genome size referring to the amount of haploid DNA contained in the nucleus measured in picograms. From this value, it was once thought that more complex organisms must have larger genomes. But in the 1970s, scientists developed the C value paradox, where there's a lack of correlation between genome size and morphological complexity. So, why do genomes in eukaryotes have no correlation between genome size and organism complexity? Over to you. Today we'll be talking to several organisms to get to the bottom of it. First we'll talk to a salamander to do a comparison of genome size and content. So tell us about your genome in comparison to a human's.
Well, I have up to 120 billion base pairs in my genome, compared to around 3.2 billion in humans. But we almost have the same number of protein encoding genes. So we have larger genomes, but are less complex organisms. This is due to our genomes containing so much repetitive, non-coding DNA, even more than humans, which I'm not complex enough to talk about.
Let's take a look at an apparently more complex organism that has a smaller genome. Can you please tell us about this repetitive DNA and the cause of the C-value paradox?
This repetitive non-coding DNA makes up for around 98% of the human genome. Most of this is intergenic DNA, such as short and long interspersed retrotransposable elements, long terminal repeats, satellites, and transposons. This DNA transposon duplicate and move around the genome, causing it to increase in size. Other contributors are gene families and also split genes where exons only take up 1.5% of the genome and are separated by large non-coding introns. This means that karyotic genes are spread further by this non-coding DNA, correlating to the large genome itself, not necessarily the number of genes or organism complexity, all of which contributors in explaining the C-value paradox.
Okay, thank you, human. Now over to a special guest that's a bit of an oddball in this discussion. E. coli, what makes you different?
I'm a prokaryote and do not fit under the C-value paradox. The relationship between the amount of genes I have is proportional to my genome size. This is due to me having almost no repetitive DNA, as my genes are more closely packed together, meaning almost everything in my genome is used, unlike the excess garbage found in eukaryotes.
Well, from this garbage, or seemingly junk DNA, we've actually learnt that it does have a function. This includes regulation of protein coding sequences, scaffold attachment regions, origins of DNA replication, centromeres and telomeres, and also teaches us about the evolutionary development of genomes over time.